Today we are recording live from Florida Agricultural and Mechanical University with Dr. Kanga Lambert, Dr. Whitley Stewart, Dr. Anamika Sharma, and Dr. Worrel Diedrick. Today we are live in Quincy, Florida at the FAMU Research and Extension Center. We ask that for those watching that you are having your gallery mode so you can see all the views today throughout the discussion. Our big themes and takeaways from today's Scientist Online Electronic Field Trip is biological control. Specifically, we will discuss honeybees, mosquitoes, and environmentally friendly ways to control pests in our homes and workplaces. All right, and Dr. Kanga, if you want to introduce yourself before you begin.
Thank you, Maggie. My name is Dr. Lambert Kanga. I'm professor of entomology at insect science. I'm also the director of the Center for Biological Control here at Florida A&M University. Now, in a nutshell, the Center for Biological Control provides biological solutions to pest problems affecting agriculture, natural resources, and human health, while rebuilding capacity for future innovations. Now, being an entomologist allowed me to travel several places, such as Africa, Asia, Caribbean, South Africa, Latin America, and so forth and so on. But today, I'm going to talk about the honeybee, which is the man's oldest insect friends. Now, the question comes, what are honeybees? Oh, they are insects, a small creature with a free body part. They have head, thorax, and abdomen. Also have 6 legs. So, we call it honeybees. But honeybees, where do they live? Several places. Natural habitat, you'll find them in cracks and crevices near trees. But domestic honeybees, or European honeybees, they live here in some of the boxes that we have, different sizes, we call wooden boxes. And now I'm going to switch over to Dr. Diedrick to talk to you about beekeeping as well as harvesting honey before I can talk about the importance of honeybees.
All right, and Dr. Worrel, if you want to introduce yourself.
Hey, Maggie. Thank you, Dr. Kanga, for that introduction. So my name is Worrel Diedrick, and I'm a postdoc researcher in the College of Entomology at Florida A&M. Today, I'm going to take you into the world of beekeeping. We're going to talk about some tools, materials, and products from the hive. So first, we're going to look at the smoker. So the smoker is used inside the hive to emit gentle smoke to calm the bees, so that to make the environment less stress-free for the bees while the beekeeper is working. And we use the bee brush inside the hive. We use a tool called the hive tool. But this tool is very important. It is used to move hive parts around, also to scrape away propolis, and it's very handy inside the hive. Next, we're going to go to the suit. Have you ever got stung? Well, as beekeeper, we wear our suit, and this is to prevent bees from stinging us. Also, gloves to protect the hands from stinging while working inside the hive. So we're going to look now at the home of the bee, where the bee lives. And right here, we have 2 different types of homes. We have a nucleus home. So this is where you start new colonies. So this is just 5 frames, and you put the bee inside and start a new colony. And as soon as that colony gets larger, then you'll move them over to a standard size, long, short, 10-frame hive, right? So we're going to look into the hive a little bit closer. So as you can see here, so 10 frames at the top, and here is where we call the super, and you can see the the queen excluder. So this is used to keep the queen in the brood chamber. So as the name suggests, the brood chamber is where you'll find the queen and all her babies. Yes. All right. So this now is our display hive. And the display hive is just a snapshot of what's happening inside the big hive. So if you look closely, you will see the bottom frame with all the baby bees, and the queen would be down here and she would be laying. Up the top is where you'll find the super, and this is where they'll store the excess honey. So this is just a small version of what you'll see here. And then you can see we have different types of bees. You have the worker, and you have the queen. So these are the most important in the hive during the all year round. You have the drone, but during breeding season. Dr. Kanga will go more into the hives. So then we're going to talk a little about pests. So we have the varroa mite, which is an important pest of the honeybee, and we use the extracticide to apply insecticide to kill the varroa mite inside the hive. And Dr. Kanga going to talk a little bit more about what we found to use in the extracticide to kill the varroa mite. So now we're going to move to the fun part. And the fun part is products from the hive, right? So one of our products is the
Honey.
honey. Everybody loves to eat honey, right? So how we get the honey from the hive, we'll go inside and remove a frame from our hive. So the frame would be filled with nectar, and that nectar would be mature and ready, turn honey and ready to be harvested. So we would use the uncapping roller, and we would remove the thin layer of wax that the bees use to protect the honey inside the frame. And after we open, you can see it have sharp teeth, prongs, right? So after we open the cells, we would put the frame inside on the extractor, and we would spin. We'll spin. So this centrifugal force will flash the honey from the frame to the side of the drum. And it would go down, and then we would use a strainer to catch the honey and remove all impurities. Then we'll put it inside the bottles, and it's ready to be eaten. Right? Have you been to Walmart get honey? Yeah, delicious, right? So not only honey we get from our hives, but we also get pollen. Pollen is a very good source of protein, and we use a pollen trap to get pollen from the hive. So come a little closer, let me show you how we demonstrate how we get the pollen. So we would use the pollen trap, and we would set it at the entrance of the hive. So when the bee coming in with the pollen sac and the legs, the trap will gently remove it from the leg into this basket here. Then we would dry that pollen and put it in the container, and it's ready to be used. So thank you for joining me today into the world of beekeeping. And now I'll turn over back to Dr.
Awesome. Thank you so much. All right. And Dr. Kanga, back to you. So bees, where do they live? And now we're on the importance of honey.
Oh, yeah. Importance of honey. As you know, honey is very, very important. Not only for honey productions, but more so for crop pollinations. So we know that each year, the honeybee industry produces about several $1 million on honey products, and about $200 billion worth on pollination of crop and vegetables.
Wow.
Some of the crops that we talked about, like blueberry, onion, avocado, and so forth and so on, and almonds, they're 100% dependent on honey for pollinations.
Wow.
And be also very important in terms of biodiversity.
Mm-hmm.
Yeah, because honey pollinate wild plants. And honey products also used in health food and alternative medicine.
Right.
So without honey, no hamburger, no tomatoes, no food, no vegetables.
That's Rough. It sounds rough.
Oh, yeah, it does.
Yeah.
So therefore, we know that honeybee is very critical for our own well-being and our survival.
Mm-hmm.
But now, now, we talk about how they live. Now we're going to see, how they grow. Therefore, I'm going to turn over to the life cycle of honeybees. And then we, as an entomologist, say, okay, the life cycle is complete because it go from eggs, and then we're going to have larvae. From larvae, you go to pupa. From pupa, you go to adults. That's a complete life cycle. Now, bees are very smart. They're social insects, so they all live together. But they're also divisional neighbors. They have 3 different castes. Yeah, the queens, so only 1 queen per hive.
She's a big deal.
Oh, she's a big deal. They can look at her belly. It's very big belly.
Yeah.
So therefore, she laid only once. And then she laid about 2,000 eggs a day.
Wow.
So therefore, the queen is very productive first 2 years.
Mm-hmm.
But she can lay up to 8 years. But beekeeper used to change the queen every year or every 2 years. Now, we have, after the drone, after the queen, we have also the drone. Those are reproductive male. They don't have stinger.
Oh.
But the only role of a drone is to mate the queen.
Right.
Now, when they don't have enough food, you know what the worker do?
Out they go.
They put them outside, and then they all die out. And then we got workers. They are the one who take care of the housekeeper. They clean the house, they fix the crack and crevices. They go out and forage, feed the larvae as well as the queens. Now, we know that there is a decline of honeybees. It's been almost over several media, like ABC, CNN, National Geographic. The bees are dying. So why they are dying? Because of several factors. But the main one is habitat destructions. So they don't have a forage that they used to have anymore. So they don't have any more food. They're exposed to a pesticide, so they die from pesticide exposure. Also pests and diseases. Now, because that's problem, you know what USDA did? They said that in 2030, if the trend continue, we have to import vegetable from China.
Oof.
I don't think they want to do that.
That's a lot of work.
That's a lot of work. And also we had to import killer bees from Mexico to do pollination services. And we may have shortage of food as well. So why they're doing? Because the small creature, this small mite, we call it Worrel mite. It's from Java in Asia, and it came here, caused weight loss of the bees, shorten lifespan of the bees, and also serve vector diseases.
Right.
So without adequate control, these mite populations, bee mortality can reach about 100% of hive, and the hive will die out. Now, you know what happened to that small creature? They develop resistance to chemicals that we used to kill. So,
typical.
Yeah. So it doesn't want to die anymore. So even if you're going to spray cement chemical, this small creature is not going to die. So in our lab, we say, okay, we're going to address this issue in 2 ways. First is to be able to determine mechanism of resistance, which is the strength of the resistant pest. And then if we do that, we can make that pest be susceptible again. And then the second way is, let's use biological ways, using fungal pathogen.
Okay.
Now, to be able to determine mechanism of resistance, we look at different enzyme and some of the protein, the functional protein. Everyone break down the chemical, therefore it becomes resistant. So those enzymes are called glutamate S transferase, another enzyme called aldehyde, another enzyme called esterase, and then we look at also misfunctional oxidase. So those are different enzymes used to break down chemicals. And then we use those fancy, those fancy equipment So we use molecular and biochemical technique to do that. Now, what do we find when we look at the activity of those enzymes? We find that first, when we talk about beta S transferase and aliesterase, they're not part of the resistance. But we have an enzyme called esterase and misfunction oxidase. They're major factor of resistance.
Mm.
And then we say, okay, let's go further. And then we find that the change does not occur on a structural gene, but a regulatory locus control the expression of those different genes. They turn on and off, expression of those genes. Therefore, we can find some chemical like formamidine, a PBO can block those enzymes and make it susceptible again. Now, in terms, also we look at the gene sequence to see, what this amplification came from. And we find that there is a kind of mutation of a gene, the replacement of some of the amino acid. That's the reason why we got those resistant. And in terms of biological aspect, we look at different fungi. So we test many of them, about over 100 fungi, and we find that two of them, Meurasium and Ursitera, can kill the mites, but harm the bees.
Perfect.
Yes. So okay, let's try this then in different states.
Okay.
Make sure that it works.
Right.
Yeah. So we did that in Florida, Montana, in Texas, in California, and we find that exactly this particular fungus works very well.
Great.
So now I said, okay, it worked. So what we're going to do with that? I said, okay, let's work with fungal producing companies.
Perfect.
Yeah, they came and said, okay, let's do it. So working on Nicodem, working also with Sylvan to be able to kind of formulate this fungal pathogen and put in the market. Now, I'm going to switch gear very quick and talk to you about entomologists, So we job opportunities, So as an entomologist, you can work at chemical companies like Monsanto, Terminus. You can work at the U.S. Department of Agriculture. You can do industry, drone, robotic, and so forth. And we also have...
Sorry again?
We also have assistantship and scholarship and internship. And, sometimes, right through, so you can, come to our university, just apply to the university. And my contact number is Lambert Kanga at FAMU.edu, and then select insect science as your major.
Perfect. A great opportunity for everyone listening here and to take note of this page for sure. And Dr. Lambert Kanga's information as well. So as we're wrapping up this section, we want to say thank you, but we also want to ask if anyone has any questions to let us know in the chat and we can answer them. So we'll give a few minutes for you guys to give us your questions and we'll hopefully answer them for you.
All right. It looks like we don't have any questions right now. That is totally okay. So we will save those questions for the end of our program. But with that in mind, that gives us more time to start with our next scientist.
Thank you so much, Caroline. All right, everyone. And we're going to get started on our next section here with Dr. Whitley Stewart. If you want to introduce yourself.
Thank you, Maggie. My name is Dr. Whitley Stewart, and I'm a recent graduate from FAMU. And we're going to be talking about monitoring for resistance in insecticides for mosquitoes, particularly Culex nigropappus.
Perfect.
All right, Maggie. So these are going to be the major infectious diseases of the world. And so respiratory infections rank number one. And then you also have malaria, HIV/AIDS, tuberculosis, and neglected tropical diseases. And the number two is going to be malaria, which is a mosquito-borne disease. All All right, so these are also the major mosquito-borne diseases in the world. So the Aedes mosquitoes are going to transmit dengue, yellow fever, chikungunya, and Zika virus. The Hemiplegic genus is yellow fever, Anopheles, malaria, lymphatic filariasis, Culex, also lymphatic filariasis, and encephalitis.
A lot of diseases.
A lot. Now, Maggie, are you familiar with where mosquitoes can be found?
Not really, no.
Oh, okay then. Well, you can find them everywhere except Antarctica and Iceland.
So it sounds like I got to move.
You got to move here and fast. And fast. And I also have a star here on Alaska here.
Mm-hmm.
And this is actually a photo of a mass emergence of mosquito pupae. And when the ice melts in May in Alaska, this is a triggered swarming event that occurs.
That looks terrible.
It looks very terrible.
Yeah.
All right, so mosquitoes that are commonly found here in the United States. You have the Anopheles quadrimaculatus. It's our malaria mosquito. Culicilla melanoria, Coquilitidia perturbans, Culex pipens, Culex quincke. Aedes aegypti is our yellow fever mosquito, and Aedes albopictus is the Asian tiger mosquito.
I don't think I can even say any of those. So that was great.
Totally understand. And so these are the diseases found here in the United States. So Anopheles, again, malaria, which has been eradicated. And you also have filarial worms. Culicilla is the encephalitis. Coquilitidia is Eastern equine encephalitis. And Aedes, you can get chikungunya, Zika virus, and also dengue. And then Culex mosquitoes are going to be your West Nile virus, St. Louis encephalitis, and also triple-legged.
Hmm.
Now, are you annoyed when you get bit by mosquitoes?
Yeah.
Yeah, they cause a lot of annoyance, And so that's where we get the loss of recreation sites. And then you're going to have a decrease in milk and beef production and also crop production. And they're known to cause death in our little pets, like our dogs, dog heartworms. All right, so these are the distribution of Culex mosquitoes. So you can find them all over the United States. Culex pipens is usually the north and western United States. Culex quincke, however, is anything in the southern United States. Culex nigropoppus is the southeastern United States. And that's the one in particular we do here at FAMU.
Okay.
So do you know any methods of controlling mosquitoes?
Honestly, not really. Not really.
All right, so you have physical control, chemical control, and biological control. Now, physical control, you're going to actually dump out rainwater. Anything that's collecting rain in your yard, you can throw that away, throw it out, especially bird baths, tires, anything like that. Well, mosquitoes will lay their eggs and have a mass population going on. Those too.
Gross.
All right, so mosquitoes can be controlled chemically. And so you have the chlorinated hydrocarbons, carbamates like Worrel, organophosphates like malathion, and then pyrethroids is like permethrin and cypermethrin. Now, we can also control mosquitoes using biological control, like the mosquito fish called gambusia. Now, we also have copepods. You can also use bacteria called Bacillus thuringiensis to control mosquitoes. They usually call them mosquito dunks at Lowe's. So that's what those are, yeah. And so with those control measures, you usually find some drawbacks, and they're going to be labor intensive. And for the physical control anyway, for biological control, the gambusia fish are known to eat our native species, the eggs as well. So that's a drawback. And then the chemical control usually development of resistance.
Right.
So what is insecticide resistance? Insecticide resistance is actually the reduction of the sensitivity of the insect population to insecticide. And so in this photo here, this picture, so in the red,
around 5:30, 6. And they were spending about 4 hours a day.
It's a resistant acid.
Bottom line was they get here from species. Because I downloaded course and with the knee pads and their tools. I even had the cover there.
Acceptable. Leaves a few FAMU leaves that resistant one. The resistant one is going to mate with the susceptible one and create more resistant populations. So we go to spray that later generation. It's going to kill off those susceptible ones, but leave those resistant ones here, right here in red. And
That's
this is
the
how
issue.
resistance can create it. Yes, ma'am. Yes, ma'am. So how do the mosquitoes combat resistance or combat the insecticide? You're going to have metabolic metabolic resistance, which is where their enzymes actually decrease the amount of insecticide inside their bodies. And then on the target site that was working for the insecticide doesn't work anymore.
Mm.
And then decreased uptake, the insect has a build up a thick cuticle, so no longer is able to uptake the insecticide like it once was. And then like behavioral resistance, the insect avoids the insecticide altogether. But they're pretty smart.
Yeah.
So here at FAMU, we study Culex nigropoppus. And it's of medical importance because of West Nile virus, Triple E, and St. Louis encephalitis, all diseases that we can get from mosquito bite.
Yes.
This mosquito can be found in all 67 counties, and there's very little research on its susceptibility to insecticides. So we need to know. We can find this mosquito throughout the southeastern United States. Like I told you, it's in all the counties here in Florida, and it likes the warm, humid coastal habitat along with subtropical areas. Now, mosquitoes, it's particularly Culex mosquitoes, they lay their eggs in raft shapes. Okay. And this particular mosquito's raft shape is like a gondola, like the boat in Paris. Okay. Yeah, pretty cool. And they like to lay those rafts in organically rich waters.
Right.
Like the really smelly stuff.
Yeah. Sounds great.
Yeah, I know, right? Delicious. Now, those mosquitoes are, as soon as they hatch out from the pupae, they actually are ready to blood feed at almost 48 hours old. So they're looking for you. Yes.
They're on the move.
On the move. On the move. And they're very opportunistic. So they'll feed on frogs, tadpoles, birds, any host that they can find pretty much. And during the spring months, that's when they feed on birds. And that's when they usually collect the West Nile virus and then causes problems in the summertime.
Right.
Now, for monitoring for resistance, what you have to do is what we've done is we've used the Kanga plant method where we've actually treated 20 milliliter scintillation vials with the insecticide mixed with the acetone. And what we do is we roll them until the acetone evaporates. And then after it evaporates, we add about 1 to 2, maybe 3 mosquitoes per vial. And then after 24 hours, we record any mortality that we see.
Okay.
And we put that information into a probate analysis. Now, from there, we come up with the diagnostic dose, which is the dose that kills only the susceptible part of the population, and it leaves all the resistant mosquitoes alive.
Okay.
So we've done monitoring for permethrin and malathion. So permethrin is going to be 0.1 micrograms per vial, and for malathion is 0.01 micrograms per vial.
Okay.
Now, from there, we were able to determine the frequency of insecticide resistance in mosquitoes in Escambia County and also Pasco County. Okay. So this first part of my bar graph or whatnot is the Escambia County in June of 2016. Now we see a 30% frequency of survival here in Escambia County to malathion, at 0 to permethrin. And the same thing happened in July. No change, still about 30%, no change. But in Pasco County in 2017, we actually found a resistant population. And it was a 90% survival rate to malathion in both months, February and March.
Big jump.
Big jump. And now from that data, we were able to recommend and give resistance management strategies. And what we would suggest is you restrict the use of said insecticide or use a synergist, and also rotate different classes of insecticides with different modes of action.
Gotcha.
Thank you so much.
Thank you so much. All right, and we're going to go into our next question segment. So if you guys have any questions on this research second section, or if you didn't get a chance to ask any questions from the first section, please let us know in the comments and we'll answer some of those.
So we do have a few questions. We will start off with Dr. Whitley's questions. We have one from, I'm sorry if I get your name wrong, but Halen. What is the most effective way to prevent mosquitoes? Are insecticides still the best?
Oh, that's a really good question. My best answer for that is to make sure that you're doing what I told you in the beginning about physical control, and you're eliminating all sources of rainwater, especially around your home. And if you can't eliminate those sources, I suggest using what I just call the mosquito ducts. You can find them at Lowe's at Home Depot, and those will effectively control your larvae population, so you won't end up with adult mosquitoes.
Okay, thank you. We also have a question from Amelia Z. Is it true that only female mosquitoes bite?
It's definitely true. They actually need the blood to mature their eggs. So it's definitely true.
Your burning question has been answered, Amelia. Charles asks, is it true that there are sterile mosquitoes to try to decrease the population of mosquitoes?
Yes, that is definitely very true. They had some released in South Florida a few years back. So yes, it's definitely true. These are good questions.
They're very good questions. Also, I don't think you heard it for that last one. We had a resounding yes from some of our experts in the room. Let's see. Tommy is asking, why do some people get bit by mosquitoes more than others? Is it the blood type?
It could be a variety of factors. Honestly, Dr. Kanga is more effective in answering that question. He understands resistance a little bit more, or he understands that a little bit better than me. I actually just asked him this. So
I'm back, Dr. Kanga. Oh, soft your skin.
Actually, that's a preference. It depends how much CO2 that you emit.
Okay.
Emit a lot of CO2, and then the mosquito pick it up and then follow you around so it can feed. So CO2 is one of the cues that they use. And
And it's a little bit about physical barrier as well. Say, for example, if you have a lot of hairs on your skin, then mosquitoes will find it easier to jab on a skin where they don't have much hair. So that's why. No, carry on.
Yeah.
Thank you.
So we're going to do a couple questions from the first session since we didn't have any at that time. So to start off from Neater, how fast do bees fly?
Fast to be fly. So bee can fly up to 10 to 12 meters per hour.
10 to 12, say it again.
10 to 12 mph.
Okay, miles per hour?
Hours.
All right.
Yeah.
10 to 12 miles per hour. That's pretty fast, actually, for a bee. Tommy asks, how do you remove beehives from the inside of walls?
So that's a good question. So if it's a wooden structure, what you would do is to first pry away pieces of the wood so you can get access to the hive. After you get access to the hive, you can go the fancy way. There are insect vacuums that you can use to suck the bee away and it won't kill them. It will gently remove them into a container where you can put them into a box, or you can do it the not so fancy way where you can use a knife and you can cut the comb and remove it, tie it to a frame, put it inside a box, and then use a scoop and a brush and get the bee and put in the box.
Are there any ways you recommend keeping the bees calm until you are able to move them?
So just use smoke, and once you use smoke and put them in that box, you just leave the box close to the area where the colony was. And the queen pheromone, she will emit this pheromone and it will, are in the workers. So all the workers will go right where the queen are inside that new box.
Okay. And then we have one more question. Oh, someone asking else?
The hard way to do it is to try to find the queens. So you can find the queens, and relieve the queen and put in your boxes, they follow up.
How do you find the queen?
Well, you look, put your hand there and look. Yeah.
Like a really hard word.
It's very hard work. But put glove and try to find the queen and remove the queens, and then they'll follow.
And then one more question, and I'll move on to our next section. From Sarah, what led to Dr. Kanga and Dr. Worrel to want to specialize in honeybees? Also, have they read the book, The Secret Life of Bees?
So I've heard about the book of The Secret Life of Bees, but I haven't read it.
Okay.
But my passion in honeybees started way back when I was in high school. So I used to do bee work as a holiday job. And then I developed a love for honeybee because apart from you can make a little money from it by selling honey, you can also get a chance to pay back to the environment. Because by keeping bee colonies and watching them going around and pollinating flower, it helps with the diversity of plants. It helps with fruits. So somebody have a vegetable garden in the community, that will help those gardens to be more productive. And so that's what drives. So it's a passion to see, it's to pay back to the environment.
It's amazing that something you're so interested in high school, like, still drives you to this day. That's awesome. Dr. Kanga, what about you? How'd you get into this?
How did I get into bees? When I was growing up, I worked in a farm with my dad, and he got vegetables. And I find out that most of the vegetables was not producing anything. And somebody told me that I wasn't producing because I didn't have enough bees. And I told Dad, it's okay. When I grow up, I'll make sure that I study bees, and then I'll use those bees for you to grow your vegetables. So when I went to graduate school, I was working with a professor who was interested in bees. And I said, okay, I'm going to specialize on those bees. So I became, beekeeper. So I work on, even though I was going to school, but I was going to help in apiary. I developed those skills and learn how to deal with bees. And I mentioned it took me almost everywhere. I went to several states, to several countries, just by talking about bees. It's a very good thing that I chose to work on bees.
Thank you so much. So we will go to our next part of the program. If you still have questions on either of the programs we've already had, we'll still take questions at the end, so don't worry. But now I will turn it back over to our host.
Awesome. Thank you so much, Caroline. And thank you guys for all your questions. Make sure to keep them coming for this next section for sure. And so our next section here is going to be with Dr. Sharma and her graduate students. If you want to give yourself a little introduction and tell everyone about yourself. Well, thank you, Maggie.
Thank you, the entire team, for having us and giving us an opportunity to talk to the young kids. With me, first, let me introduce my postgraduate students. With me, I have Jacques Daniels. He just started his graduation, and he's working on black soldier fly. And then I have Wesley Sentilis. He is working on IPM traps, and that's the one topic that I'm going to talk about. And Jacques has earlier worked on IPM traps. So with that, my name is Anamika Sharma. I'm an entomologist, insect ecologist. Currently, I'm working as an urban entomologist. That means I deal with urban insects. And my passion is in biological control and integrated pest management. Does not matter in which area it is. So I think that's a good enough introduction.
Yeah, it sounds great to me.
Okay, so before I talk, let me give back bring few things.
Mm-hmm.
I know Dr. Kanga has spoken about resistance.
Yes.
Dr. Stewart has spoken about management of mosquitoes.
Mm-hmm.
Some of those topics will come in this topic as well. So I hope the students will be able to relate to it. If not, then of course they can ask us questions.
Of course.
So let's talk about the chemical use in urban areas. We will talk about what urban area is, but what is chemical use in urban areas? Urban area is all we live here. So currently 30 to 40% of pesticide use That's the entire use throughout whatever system we are doing it. It is in urban areas.
Okay.
And this is no surprise because we don't want to see insects close to us, any arthropod close to us, because we consider that all insects are pests, which is not true. There are only few, very small percentage of the entire insect group is pest.
Right.
So we need to learn to manage our pesticide use. And that's the target of reason behind using the IPM traps.
Correct.
That is why, because this is so much of insects around us, that is why the urban pest management industry is $1 billion pest management industry, because no one wants to see a single cockroach in their house.
No, I sure don't.
We constantly are spraying, and that's why it's $1 billion industry. How does it impact other than the direct spray? When we spray pesticides in our lawns, in our backyards, in our houses, it's not just spraying and getting direct exposure, it's about water runoff as well, because there's so much water runoff that happens, and then that brings us to more hazardous impact of pesticides. That is why it is very, very necessary for us for us to reduce the pesticide used in environment.
Right.
Because I'm going to talk about IPM traps, so I'm going to briefly introduce the term IPM to our audience. So IPM is, the full form of IPM is Integrated Pest Management. Again, emphasis on pest. Not every insect is a pest. Please be mindful.
Right.
So what we do is that we integrate a lot of components so that we can reduce the use of pesticide and integrate other strategies to manage pests. That is why, if you see on the slide as well, you will see we have environmentally friendly, socially acceptable, economically practical, because it has to be financially profitable as well.
Right.
And then overall goal is how to reduce pests. Okay. With that, let's briefly talk about urban landscape. As we have spoken about already, any park, garden, apartment complexes where we live is urban. And I was informed that all the audience, the 4-H kids we have in this group, they all are the urban audience.
Yeah.
So they, of course, know what urban landscape is.
Yes.
What is urban farming? Any farming that we do, any produce we are producing in our backyard, close to an urban environment, is urban farming. Hydroponics, aquaponics, poultry, even beekeeping. A lot of urban farmers, even they have 2 or 4 beehives. A lot of urban farmers have beehives as well on their backyard. They do composting as well. So that all falls into urban farming. And they also produce the small goods as well. For example, jams, pickles, honey, that all is produced by them. That makes us very, very necessary that our urban farmers are more organic. But it is not that easy, because when we want to manage a pest, we want to manage a pest. We don't care if we are using the conventional pesticide, right? If you have mosquitoes, you're not going to think that, oh, I have to use some environmentally friendly method.
Right.
You're going to pick up the pesticide bottle and spray it, right?
Exactly.
So that is why we need to incorporate few strategies. I think young kids should also be aware that these are the strategies that we can incorporate. I am pretty sure that all the audience that is listening to us today, they know the IPM traps. They have used it. They have seen these traps in. But I'm going to talk about how do they work and how effective they are.
Perfect.
Okay. So what is IPM trap? You use any environmentally friendly component with any trapping method, that becomes IPM trap. For example, it could be light, it could be heat, it could be growth regulators, it could be pheromones, it could be conventional pesticide as well.
Right.
Because putting that in a small quantity and a small trap is going to reduce the pesticide use overall. And our target of IPM is reduce the pesticide use, right?
Right.
So straight to the traps. First of all, let's talk about sticky traps. Sticky means, as name suggests, anything that can stick. So it's a sticky. That calls the sticky trap. Means if you put it out and if insects can get stuck on that, that's called sticky trap. So now I'm going to ask Jacques to show us some sticky traps.
Yeah, so as we have here, when it comes to sticky traps, not only do you have monitoring, you have killing of the insects as well. So see, these are some of the traps that I collected and put out last semester in my undergraduate research. So the way it works is you basically take these traps here, you will pull this tab down right here, and these are basically 2 different traps separated. So you would have these 2 traps here, and you could basically put those traps anywhere in any urban environments in the kitchen, kitchen, on the counter, your bedroom, on locations like that, anywhere in the house, in the garage as well. We also have some traps here that have attracted non-targeted pests as well. So you have some geckos here, roaches as well, have some centipedes and beetles. These traps here are actually specifically for German cockroaches. So these have pheromones that attract both male and female cockroaches. These have pheromones, and Dr. Sharma will tell you more about pheromones in the next set of traps.
Thank you, Jacques. So that was our sticky trap. Location is very important, like Jacques has indicated. We have to make sure that the traps are at the right place to make sure that they are effective enough. Okay. Now imagine that you have a sticky trap and now you add another component that is light. So that becomes light sticky trap. For example, a lot of household insect pests are attracted to lights, right? Mosquito, flies, they all are very attracted to light. So this is one example of that sticky trap that has light component included in it. And now Wesley can a little bit show that how it works.
Yeah. So here we have the silent trap or flying insect trap. All you have to do is plug it. It's electronic. And then we have this card. You just put these cards in. When you plug it in the light, you will attract the insects in. And once the insect enter, it grab it. And as you can see, we have these. We have a lot of insects.
A lot of them, a lot of beetles in there.
That's right. Right.
So it's about, can we monitor presence of insect? Do we have to spray the pesticide? If the insect is not present, then we don't have to. So trap work in two ways. One, can we monitor? Can we find out if the insect is present? Second, when they get trapped in so much number, we are killing the population as well, right? So we are reducing the population. Okay. Now, going to another kind of traps is pheromone traps. Like we have already indicated, there are pheromones, right? What is a pheromone? Pheromone is just like hormone. The only difference is pheromone is secreted outside the body and it doesn't impact it actually impacts the member of the same species of that organism. For example, human pheromone is going to impact other humans. Right. A fly pheromone is going to impact other flies, right? So, and why do insects use pheromone? They use for several reasons. They can be alarm pheromone, means they are alerting that there's a danger here. Or it could be a mating pheromone where they are calling each other, male and female, for mating, means more bugs. And we want to avoid it. Right. So what we do is that we sometimes create the mating pheromone. So what will happen is that when we put that thing, this kind of place, which is very, very safe for both, pheromones are very safe for both humans and pets. So you can very safely put that in your pantry, close to your food as well. Perfect. And then when it comes out, the pheromone, they're going to attract the particular males or female, depending on that. And then they will get stuck there. Again, two things. We know the presence, so we are monitoring and we are killing as well. Right. So reducing the population. And then this is another kind of pheromone trap that we have here. Different styles are created targeting the different insect. If you are targeting a moth in your pantry, then a different kind of a style. If you're targeting a beetle in your pantry, then a different kind of design. Right. Okay, so now I'm going to talk about the next kind of trap. I hope we are not going too fast, but we have to. Okay, so this is a termite trap. All the kids are from Florida, right?
I believe so.
Okay, so if you are from Florida, you know termites very, very well. It's again, like I said, it's $1 billion industry. Oh my God, you have to do pre-treatment, then post-treatment. You have to constantly watch for termites when you are a house owner. You don't want to lose your house. So we have this baiting system. So far, as of now, as of April 2024, this is the best method to take care of termites. So what we have here is that this is a trap, which we put that in ground. We dig about a feet deep hole, that this in, and depending on what is the perimeter of your house, you can put as many traps as you want, depending on the area. Usually you target 10 to 20 feet distance between one trap to other. And what happens is that in this one trap, you have bait. What is bait? Anything that can attract termite. For example, this is a very tasty bait for termite, more than the wood in your house. So when we put these termite baits in ground, what we are actually doing is that we are asking termite not to go to house, rather come here.
Sounds good to me.
Yeah, because we have more tasty food here. Right? Yeah. So, and what do termites eat? Cellulose. So we give them a really tasty treat here. They come here, they get trapped here. That's monitoring. And if we can add an insecticide here, which we usually do, that insecticide is a chitinase synthesis inhibitor. And in easy words, what it is it reduces their molting process. That's why it reduces their population. So they come here, they get attached by the pesticide, what we are using here. They go out and transfer it to their friends as well, friend and family. And it's actually one family because termites are social insects. So they give that insecticide to their family member and voila, you're good to go. Like I said, although we are using conventional pesticide here, but we are not madly just spraying everywhere.
Right.
It's in a small container. So less harm to environment, less harm to humans, and less harm to pets, less harm to everyone. Okay? Win.
All over.
And here are some biopesticides. So same, imagine that I'm using this trap, and instead of using a conventional insecticide, what if I'm using biopesticide? What is biopesticide? Any chemical that originated or produced from a living organism. It could be a virus, bacteria, fungus. Like in earlier talk, Dr. Kanga has spoken about the fungus that he has used. And so anything that is produced from a living organism is called biopesticides. Now imagine instead of using a conventional chemical pesticide, if you use biopesticide in it, then it's a super win situation for us, right? It's absolutely environmentally safe, very safe, right?
Great.
That's why we use biopesticides there. I'm sure a lot of kids, they have seen this, the bait. It has a spinosad in it, and it can kill fire ants. We all know what fire ants are, right?
Yeah, no more fire ants.
No more fire ants. But the efficacy could be a problem. So I'm going to talk briefly about it, the limitations of biopesticides. The list that I've shown here is just to indicate that there are so many products available to kill ant, but there are only 2 that are biopesticides. That means more research is needed.
Right.
There are a lot of gaps that we don't know. That is why more young people are needed.
Yes.
And that's when you can make a difference.
Yes.
So briefly, what do we have to do if we really want to incorporate IPM strategies to managing our household pests? We have to stack a lot of management strategies, including you eliminate their food, water, and shelter. If they don't have it, they're not going to come to your house. For example, if you have cockroaches, make sure that there is no leaking water there. Don't keep your containers open, and always close the small gaps that you have close to your several things. If you do that, you're going to save a lot of trouble to yourself. Okay. And most importantly, talk to each other.
Yes.
If you have a pest, talk to other person. Do we have a pest? What are you doing, right? And so communication and being aware of a presence of a problem is very, very necessary. And that's when you can, if you know that this is a huge problem, you can go and talk to the expert and they can guide you better.
Right.
Rather than grabbing a pesticide, spray that from a grocery store and then just spraying it madly.
Right.
Briefly, I'm going to talk about some success stories.
Okay.
One of them has mosquitoes. I know a lot of people were interested. They asked Dr. Stewart as well that how to control. So this is a success story of mosquito trap. Again, the insecticide that is used in it is a conventional insecticide. But the good thing is that it's not sprayed madly like fogging for mosquitoes.
Right.
It is there in a small container, so it is not exposed to pets, to humans, to environment. The red and black color attracts mosquitoes. And the insecticide that is used in it, the small strip, the white strip that you see there, that can kill both larvae and adults. All you have to do is that you put that in your backyard and then mosquitoes will come there. So you are eliminating the backyard mosquitoes without spraying any insecticide.
Right.
This is another success story that I showed you, the termite trap. This is a success story because like I said, this is the only trap we have, baiting system that we have that can manage termites comfortably and feasibly. It's a feasible method to manage and it is more environmentally friendly.
Right.
Another example I have, I don't have this with me, but it's a success story. This is a biopesticide trap for or the spray for bed bugs. It uses an insect killing fungus.
Okay.
It is developed by one of the scientists in Penn State. Then, like I said, biopesticides and IPM traps, they have a lot of challenges.
Right.
We don't know much about them. We don't know how effective they are. In general, if you ask people, they say, oh, no, they don't work. They don't know how effective they are. So a lot more research is needed. A lot more young people are needed.
Yes.
And their shelf life is small, so more work is needed to increase their shelf life. Okay. Then the future is automatic trap. Future is for AI, right? We all know what ChatGPT is by now.
Yeah.
So now the automatic traps are there. You install a trap and you go anywhere you like, and you can look at your mobile and you can figure out, okay, do we have a pest or not? Even the pesticide applicator company people, they can install a trap, 1 or 2 traps in your house, and they don't have to constantly come and knock your door.
Right.
At their places, they can see and call you, oh, you have this problem, right? Look at that.
Even has its own Wi-Fi.
Right.
Look at that.
Exactly. And then, but again, a lot of research is needed. A lot of pictures, ID, everything has to go in system so that this piece of equipment can identify correctly.
Yeah.
Correct identification is very, very necessary.
Yeah.
You don't want to have a cockroach and spray for mosquito, right?
Yeah, exactly.
So pesticide resistance is not our fault, but we are part of the solution.
Yes.
And with that, I'll say thank you very much.
Thank you. And once again, we're going to move into our next section of questions, our final section of questions. So if you guys have any questions specifically for this section or the other past sections, please let us know in the chat and we'll get Caroline rolling them out to us. Oh, you need the microphone. Sorry, everyone.
Okay, so we do have a question from Sarah. How can students stay updated on educational and career opportunities at FAMU related to entomology? Was it Dr. Kanga? That might be a question for you.
And Caroline, could you just repeat that question?
Yes. How can students stay updated on educational and career opportunities related to entomology at FAMU?
So first of all, we have a website.
Okay.
We started working on it a few months ago, and it is being established. So you will have all the opportunities indicated on the website. Second, we will post some flyers here as well, so you can get our contact info. And if you contact any one of us, the faculty, we will give you all the information.
Okay.
Here, come on too. You talk about the classes you're taking and what you're studying and working on as well. I think you're actually loving to hear from some current students.
Okay.
What you're all studying and working on, some of the classes you're doing, why you chose to come here. That would be great.
Yeah, I'd be more than happy to share my experience. So I kind of always known when I was going to college, entomology was my thing. I always knew I wanted to work with insects. So my undergraduate, I did focus on these trapping systems that you see here, urban entomology. Here in my graduate years, I'm focusing on black soldier flies. They're good for agriculture. A lot of times the larva is used for feed. So that's what I'll be focusing on in my graduate studies. Some of the courses I've taken have been pretty interesting. So I've taken forensic entomology in my undergraduate years. So those who are interested in CSI and investigations and things of that sort. So you have your bodies and you can see the different insects that are on the bodies and the decaying time and things of that sort. I've taken general entomology, so you get your basic understanding of the life stages and things of that sort. I've taken insect morphology, so you're looking at like the characteristics of the insects, the wings, the legs, different variations, the different eyes and antennas that they have. So there are a lot of really good courses that are involved in entomology.
I'm Wesley Setilis, and I'm international student. When I was in my country, I was really interested about insects like honeybees. But when I have this opportunity to come here in FAMU to study entomology, to be graduate student, that was so amazing to me because I love it and I enjoy, work with insect and then enjoy when they fly in the nature. That's so amazing to me. That's why I love it and enjoy it. Thank you.
Okay.
One more question from Sean. Are the electronic net traps not considered IPMs? And if they aren't, is it because they don't use pesticides or biopesticides like the other traps?
No, they are considered IPM.
They are considered
It doesn't
IPMs.
have to be a biopesticide or a pesticide. It could be a physical thing as well. For example, heat, like I indicated. So if the electronic one are using electricity, yes, that's an IPM. Because the overall goal of IPM trap is to reduce the conventional pesticide use.
Okay. Sean, I hope that answers your question. While we are here, does anyone else want to unmute and ask a question or put one last question in the chat before we wrap up?
Either we have done a marvelous job that they understood everything, or we have done a bad job.
I think you guys have done a marvelous job. Okay.
All right. And if no one has any more questions, we want to thank everyone for joining today. Thank all our participants from FAMU for presenting. We hope you learned a lot today and share the information that you learned. And thank you once again. This is Insect Investigators.